Hello everyone! My name is Joslyn McKlveen and I'm an undergraduate student at the University of Georgia studying economics and business analytics. I'm a research assistant in the Computational Plant Science Lab and am sponsored by the Center for Undergraduate Research Opportunities. I work with Dr. Alexander Buchsch and Momo Shi. My role in the lab is to analyze the data collected from the greenhouse to determine how various environmental factors affect common bean growth, yield, and their micronutrient concentration in an ongoing experiment. Our experiment studies SEQ7 common beans, which are a staple crop in Eastern Africa and Latin America. Many rural communities heavily rely on their protein and micronutrient contents to fulfill their hunger levels and nutritional needs. We are growing common beans with both well-watered and water-stressed conditions to understand the variation of common beans' biomasses, seed weights, and micronutrient contents in relation to their architecture types. This will allow us to determine what can be done to breed for the most optimal, nutritious common bean, especially with climate change and drought-like conditions varying across different regions. To establish a baseline of the common beans' growth under both well-watered and water-stressed conditions, we measure the common beans' temperature, humidity, and soil moisture levels from February 26 to May 26. From February 26 to April 3rd, all the common bean plants were well-watered. Soil moisture measurements were taken at two different depths, with one at 6 inches of depth and the other at 15 inches of depth. Here you can see the images of the greenhouse each day, while the temperature, humidity, and soil moisture levels fluctuate. From February 26 to May 26, the average temperature ranged from a low of 20 degrees Celsius to a high of 28 degrees Celsius. The average humidity ranged from a low of 33% to a high of 78%. The soil moisture average percentages remained relatively consistent with the soil moisture levels between 29.7 and 32.0% for 6 inches, and 32.0 and 34.9% for 15 inches of depth. Starting April 4th and continuing to May 26th, half of the common bean plants were water stressed and half were well watered. As you can see, the gap between the soil moisture of the well-watered plants and the water-stressed plants grows larger as time progresses. Next, we took biomass measurements of the common beans' plants' roots after they were extracted from the ground and dried for 72 hours at 60 degrees Celsius. We did this in order to compare biomass levels between the well-watered and water-stressed plants, and we found that the well-watered plants tended to have much higher biomasses than the water-stressed plants did. The next step in my analysis was to look at the seed number. Here you can see that the well-watered plants produce much higher quantities of seeds than the water-stressed plants. The next question was to address how these differences in water levels affect the common beans' micronutrient levels. We sent off 71 common beans to a lab for their micronutrient contents to be measured for 12 different micronutrients. I split the data into well-watered and water stress among each of the 5 different architecture types that we determined, with the architecture type on the x-axis and the micronutrient content on the y-axis. The first observation I made was that some micronutrients have higher concentrations under a particular environmental condition, and that would be across all of the architecture types for that particular micronutrient. So for example, you can see here that calcium is more prevalent under water stress conditions. The same goes for magnesium in this graph. In others, micronutrient concentrations were higher under well-watered conditions. Sulfur is an example of this. Here's the graph of boron, which is another example. The rest of the micronutrients have inconsistent patterns where some of the architecture types have higher micronutrient accumulation than others. Also, within each micronutrient, it varies whether the well-watered or water-stress conditions have micronutrient contents that are higher across different architecture types. I created this diagram to try to understand the relationships between architecture types and environmental conditions, as well as genotypic variation that could be accounted for. We are still investigating these trends and using statistical analysis for more accurate categorizing. But for now, this is just a way to see the overall trends and kind of what's happening and what we should be looking into. Thank you so much for watching the summary of my research thus far. You can use this QR code to visit our lab website to see what my research lab mates are doing. Follow my social media to watch for updates on my research project. Thank you.